Now we go to the final group, our genetic destiny. In what way is the gut a gateway to altering our genetic destiny? Remember the Time magazine cover from January of 2010? Why your DNA isn't your destiny? The new science of epigenetics reveals how the choices we make can change our genes and those of our kids. Wow, that's a headful, as all new information is. And I have to drill down and see what that means. I thought it was just bad genes. The mother gives me a set of cards, my dad gives me a set of cards, and I have to play the cards I'm dealt. When we had Dr. Deal in Rockford, Illinois, teaching the CHIP program, we would say, no, you can ask for a redeal. Sorry, yeah. So, back to this tree diagram. Fundamental clinical imbalances like upregulated inflammatory responses then be connected to our protection and defense system, which is turned into a leaky gut because we don't eat right, we kill off our good bacteria with antibiotics, we don't replace them, and we don't provide butyrate to the lining cells. And that relates to this fact that our genes are turned off, all of them, 95% of the time. And they get turned on from cell signaling from food. Food can act as a cell signal. From bacteria, which can act as a cell signal. From stress and hormone action, that can act as cell signaling. So, as we work our way down to the root cause of disease, we do have a genetic predisposition, but it is modifiable in the roots, as we learned from another shout out for David Montgomery. We learned that the microorganisms, radiation, nutrients, trauma, physical activity, psychosocial stress, all these things can be root causes of these changes. A refresher course now on genetics. Everybody knows about the double helix design of our DNA. Sequencing of human DNA was celebrated a decade ago, and we'll cover that. And genomics is the study of genetic expression, but the epigenome on top of the genes modulates or alters the rate of their being turned on or turned off, and they're mostly turned off. Here's Watson and Crick in 1962. I graduated from high school in 1962 and learned that what I had learned earlier in biology wasn't all correct. They're receiving the Nobel Prize in medicine for that discovery. I believe that one day Dean Ornish will be nominated for the Nobel Prize in medicine if I have to do it myself. Francis Collins, who's the director of the NIH right now, the National Institutes of Health, running all of our nationally sponsored research, was the director of the Human Genome Project from 1990 to 2003, finding out that we have about 25,000 genes in our 23 pairs of chromosomes. And if we look at the nucleus of a cell, remember that from high school biology? A cell, and it's got that black dot in the middle, in this case a red dot with a blue center. Inside there, if you look closer, are the blue dots, which are the coiled up chromosomes, the chromatin material. Now hang on to your hat. If we pulled out one of those blue dots, you can see the double helix coiled up in there. How long do you think it is? 3 meters. Imagine something as long as a yardstick coiled up in a dot that you can see under the microscope in the center of a human cell. To manipulate a gene within that 3-meter long set of chromosomes, you would have to have special tools, nanotools, histones, and chaperones. How does that work? If we uncoil a chromosome, the first coil looks like a telephone cord. Remember those old telephone cords that would pull out halfway across the room? And if we look at that closely, it uncoils into the double helix. And if we look at that closely, we come to this series of circles, and you see a little red segment in one of these blue circles with a purple lining edge onto it. That little red segment is a gene, and it is inactive unless it is acted upon by a histone or some other mechanism that causes it to become uncoiled, and that little red segment is then free and out in the open. You see that on the right-hand side of the screen? It's uncoiled, and there's that string with the red section open. That's when a gene is active. And so these things can get turned on and turned off. In the upper left corner, it says epigenetic mechanisms, development in the uterus and childhood, environmental chemicals, drugs and pharmaceuticals, aging and diet. And it can affect, on the right-hand upper corner, health endpoints, cancer, autoimmune disease, mental disorders, diabetes, and so on. So we need to drill down further and know how all of this works. And some of you might ask, what does it have to do with the GI tract? Well, we're going to go to a researcher at a major university center. Here she is with her dog, and let her help us understand how it works.
Hi, I'm Dr. Dana Dolinoy, a postdoctoral research fellow in the laboratory of Randy Jurtle at Duke University. In our laboratory, we study epigenetic gene regulation, or how environmental exposures interact with the epigenome to affect long-term health and disease. So today I'd like to introduce you to two agouti mice, and as you can see, the yellow mouse is quite obese, and she is also prone to diabetes and cancer. But on the other hand, the brown mouse remains slender and lean, and also has a lower risk of developing disease. But what's really amazing about these two mice are that they are genetically identical. They are two identical twin sisters from the same mother. So what makes them look so different? It turns out that there's a second genome called the epigenome. Epigenome literally means in addition to or above the genome. And while the recently completed Human Genome Project identified approximately 25,000 genes, these genes still need instructions for what to do and when to do it and where to do it. And that's where the epigenome comes into play. A useful analogy is to think of the epigenome as the software that directs the genomic hardware of a computer. All of our cells contain the same DNA and genes, but it is the epigenome that decides how these genes are expressed and determines how a cell becomes a heart cell, a liver cell, or even a hair cell. Epigenetics consists of molecular switches and markers, such as DNA methylation, that help control gene regulation, in which a quartet of atoms, called a methyl group, attaches to DNA and shuts down genes. And as you can see, the red balls here are attaching to the DNA and turning off the gene. So back to the agouti sisters. In the yellow obese mouse, the agouti gene is unmethylated and turned on all the time, while in the brown mouse, the gene is completely methylated and shut down. There are also other mice that appear mottled, in which half of the cells are methylated and shut down, and the other half are unmethylated and turned on. And these mice appear to be yellow and brown. So the co-colors of these agouti mice acts like a sensor for the amount of DNA methylation present. We use the agouti mice to study how maternal nutrients and environmental factors affect the epigenome. Specifically, we wanted to know whether a mom's exposure to a contaminant found everywhere in the environment can alter the fetal epigenome and eventually the long-term fate of her offspring. In this study, pregnant mothers were exposed to a common chemical found in certain plastics. This chemical is called bisphenol A, or BPA for short, and it's present in many commonly used products, including food and beverage containers, baby bottles, dental sealants, and the lining of food cans. About 4 years ago, the CDC studied approximately 400 people, and in 95% of these 400 people, they measured detectable levels of bisphenol A. And when we fed the pregnant mothers the mice BPA, we noticed that the number of offspring with the yellow obese coat color increased dramatically. And we also saw that maternal exposure to this chemical decreased DNA methylation in the offspring and turned this agouti gene on when it is supposed to be off. So we started a second study in which pregnant mothers were exposed to BPA plus nutritional supplementation such as methyl donors like folic acid, or genistein, which is a common ingredient found in soy products. The level of soy that we provided is similar to what a person who eats a high soy diet or an individual living in Asia might eat. And once we did this, we observed that the offspring were no longer predominantly yellow and more obese, and that there were more offspring with the slender brown coat color phenotype. This indicates that maternal nutrient supplementation can counteract the negative effects of exposure to that chemical. Additional thinking about human health and disease is that it is affected by genetics and the environment. And whenever identical twins have different disease status, this was often attributed to the environment or different behavioral choices such as smoking status. But with epigenetic gene regulation, we can see that we can no longer say whether genetics or the environment have a bigger impact.
So, the upshot of all of this is that the genetic code is a gift from the past, but we can modify the gift during our lifetime, and during pregnancy, procreation, we have the chance to give our children a switch setting that is more advantageous. As you will see in this study on the left, the mother rat is eating her high-fat meal, and in the womb, the infant is being affected at chromosome 3 with an increase in proliferation or neurogenesis of certain cells, more than would be generated in a healthy diet, and that causes, as the cells migrate to their location of activity, increased peptide expression in the brain that governs or does switch settings for the activity of the offspring. So at weaning, the adolescent offspring has elevated blood lipids, increased food intake, fat preference, early puberty, and increased body weight. So we begin to see how the gut is a gateway to altering genetic expression to the degree that it's in our control. And if we look at infants, humans, if they have a vaginal delivery, they're going to have a predominance of lactobacillus, you see down the list there. If they have a cesarean delivery, different bacteria will dominate. And with breastfeeding, you're going to have bifidobacter dominance, and with bottle feeding, a different set of bacteria. So we set children up early on, whether if we put them on a brand new carpet, and then we get a baby blanket out of the dryer and put it down so they're not exposed to the dirt on the carpet, and then we don't let them have a dog, we deny the training of the immune system. And if the switch settings are off in the beginning, then it's really a lot of work to get them reset as opposed to the way we grew up in nature. Now, I'm not an expert in all of this, but this was published in the American Journal of Gastroenterology, so I guess I better keep up on it. But these are the kinds of things that we need to be tracking now in order to do our work of being educated, experienced, and as Olson said, Gwen Olson, become an agitator for support for change in these systems. Lean mice have one kind of bacteria, obese mice another kind of bacteria. Low-fat, high polysaccharide, carbohydrate diet nourishes one type of bacteria, the Western diet, a different kind of bacteria, and we now know that it makes a difference. I would ask you, particularly you who are in medical practice in some way, the special issue of the Journal of Gastroenterology 1 year ago this month, May 2014, the entire issue is on the gut microbiome in health and disease. And my thanks to them for help in preparing this talk. And again, a shout out to Susan Bloom. And finally, self-care checklist, what I've come to support. Stop the whites, white flour, white rice, white sugar, high fructose corn syrup. Avoid junk foods. Read the labels. Look for chemicals, dyes, anything that you can't pronounce. It's not good. Change sugar drinks to tea and water, and occasionally beer. Eat plant foods as grown, seasonal, and organic. Do you know, we learned a little bit of this from David Montgomery today, about plants having their defense systems. And if we eat seasonal plants in our region, they help defend us against things that we need to be defended against in our region in that season. And the same with local honey. The honeybees sampling the flowers in the region where we live are going to produce a honey that is important to us in our immune system. 45 grams of fiber a day, I usually say 35. I don't know how that slipped in there. Omega-3 fatty acids. And if you want to be all plant-based, then sea algae, DHA, is a great source. Limit caffeine, alcohol, meats, particularly cured meats, walk 10,000 steps a day, or other exercise. Develop a stress management skill so that you can convert yourself from living in your sympathetic nervous system, fight or flight response, to living in your calm and negotiating and loving and appreciating nervous system, the parasympathetic nervous system. Develop or spend time with people who make you feel good. When you spend time with people that make you feel bad, that's a toxicology in our environment. And so we can do that. And adopt, you know, if we're getting back to farm to table, regional food. Let's go one step further, as we learned from an earlier lecture, become an earth to ecology to farm to table, and that will take you all the way to health and vitality. A shout out to all these people that took me from a traditional physician who wanted to be a researcher and a clinician and a teacher to a holistic gastroenterologist. There they are, and I've added Joel Furman and David Montgomery because they became on that list today. Thank you very much.